Good evening, everybody. My name's Ian Walmsley. I have the privilege of being the provost here at Imperial College London, and I'd like to welcome you all to this evening's events, and I hope festivities in some way. Yes, there'll be a reception outside after the talk. We are here for a lecture, but we're also here to celebrate the 75th anniversary of the founding of our Silwood Park campus. So there'll be a brief interlude on that in a second, led by Guy Woodward, who's the deputy head of department out there. But first, we will hear the Ernst Chain Lecture. And Sir Ernst Chain was a major contributor to life sciences, of course, in general, and a leading figure at the college in this area. He's the winner of the 1945 Nobel Prize, together with Fleming and Florey, for his discovery of penicillin. And he, as a figure, he is instrumental in the founding of the department. He returned to Imperial in around 1960 to take up a position here, utilizing the facilities for chemical microbiology and biochemistry that had been funded by the Wolfson Foundation. Wolfson continues as a major partner for us today and a supporter of the college, and for that we are very, very grateful. Now, our speaker today is also one of the leading figures in life sciences, Professor Hopi Hoekstra from Harvard, where she is a professor in Harvard College and also the Curator of Mammals at the Museum of Comparative Zoology. I wish we had one of those. I guess we have next door, don't we, the Natural History Museum. She is a very well-known figure in her field and has won numerous medals. I won't try and mention all of them, but noting that she was a Howard Hughes Institute investigator and has won the Lounsbery Medal of the National Academy of Sciences in the US, of which she is also a fellow. She will talk today on a really intriguing subject, which I found just the title fascinating. It's about color in animals, how it's made, used, and perceived. And so I'm hoping that as an optical physicist, I will also gather something from that, if not the most profound piece is part of it. Before we turn to Hopi, and welcome to you, and thank you very much for giving this lecture for us, we're going to see a brief video that Guy is going to show us celebrating Silwood Park, one of the extraordinary pieces of the college, which was instrumental in the foundation of ecological epidemiology, which has arguably set the tone for that field, not only in the UK, but around the world. And I think it's a piece of college history of which we can all be very proud. So first, let me hand over to you, Guy, for that introduction.
Thanks very much. Thanks very much, Ian, and welcome, Hopi, and welcome, everyone. It's great to see everybody here. It's really fantastic to have a packed room and having some proper in-person interactions after the last couple of years. I'm going to give you a very brief introduction to Silwood Park, and then we'll play a short video. It's only a couple of minutes long, but we do have a couple of more extended versions, the sort of redux options, I guess, which are in total about 10 minutes, which give us slightly deeper insight into the research and teaching that we do at Silwood Park. The first one that we're going to show now, though, is really just a very, very short taster, if you like. We're celebrating the 75th anniversary, so Silwood Park was bought by the college just after the end of the Second World War, and a lot of the early research focus in those early days was on things like food security, understanding how pesticides can be used to improve crop yields, etc. And over time, what we've seen is that the focus and the range of disciplines covered at Silwood Park has really expanded, and yet some of those initial concerns that were present 75 years ago, we're actually still revisiting again now. So it's really our environmental hub for Imperial College. It's the home for about a third of the Department of Life Sciences, but there are also other aspects and disciplines within college that interact with us, and we're increasingly looking to expand and become more multidisciplinary by building links with engineers, with medics, business school, etc. So it's not just a life sciences campus, although it's the home for our ecology and evolution science. And that's really the theme that we're going to see today when Hopi gives her talk. So I'll just switch on to the video now, if that's possible, for a couple of minutes, and we'll send the links through for the longer ones afterwards for you.
Every aspect of our society, our economy, and our culture depends on the biodiversity that we've co-evolved with for our entire history. But the decisions we make now at the precipice of the sixth mass extinction are going to determine the future.
We're losing biodiversity at an alarming rate. What we do here at Silwood Park is study the consequences of that biodiversity loss.
Every person here, there's hundreds of us, master's students, PhDs, postdocs, faculty, we are all engaged in tackling some big environmental issue. And that means you can't have a conversation without learning something new. It's a great place to work.
There's a tremendous variety here of natural habitats that we can use for experiments and observations. We've got a lake, we've got woodlands, we have grasslands, we have arable fields. Everything you would need for doing productive field experiments.
There has never been such large-scale interest in restoring natural ecosystems, and that's because we recognize that there are allies in fighting climate change. But if we're going to do that, if we're going to harness this moment in order to restore ecosystems at large scales, we need to do it right. We only get one chance.
The grand challenges facing the natural world and the human societies that depend upon it are becoming more diverse, more intense, and more complex, more difficult to understand. So we need large-scale, field-based systems like Silwood Park to really road test some of the theory and the models that we're developing in the real world.
I feel really privileged to be here. It's an honor to work with the colleagues that I have. It's inspiring to see how these huge societal issues are being tackled from so many angles by so many bright and really motivated people, especially getting to interact with the students that we have here who are going to be carrying this challenge forward into the future. It's a wonderful part of being here.
Thanks very much. Okay. So hopefully that's given you a bit of a taste of some of the things that we do. As you can see, it's a rather different looking campus from the cosmopolitan campus that we're in today. For those of you who are not actually at Silwood already, who've come from outside from our other campuses, please come and visit us. We're always happy to host you. And I'll hand over now to Hopi for the main event.
Okay, can everybody hear me okay? Sounds good. Okay, thanks. So first I want to thank you for the invitation to be here. It's wonderful to give this prestigious lecture and to help celebrate the 75th anniversary of Silwood Park. When I was invited, I was given a charge, that is, can you please give us a talk that will appeal to high school students all the way up to optical physicists? So I've done my best, so hopefully there's a little bit for everyone in here as I tell you a little bit about nature's palette. So today what I'd like to do is start with an introduction to sort of motivate the work that I'm going to tell you about today. And then for the majority of the talk, I'm going to focus on a body of research that we produced from our lab, trying to understand how and why color evolves in the wild, pulling examples from diverse species as well. Okay. Trained as an evolutionary biologist and as an American coming to England, I couldn't resist but starting with Darwin. So Darwin, as you all know, in 1853 wrote his, in my opinion, most famous book on the origin of species, in which he outlined in staunch Victorian prose his theory of evolution by natural selection. And to me, what's so appealing about this theory is its elegance and simplicity. It rests on three main tenets. That is, that in a population of individuals, they compete. Those individuals compete for a limited number of resources. And if there's variation in their ability to compete for those resources, and the traits that affect their competitive ability are at least partly heritable, then that population should be able to evolve, to adapt to new or changing environments via the process of natural selection. Now, in Darwin's tome, he carefully documented through observation and elegant experimentation data for the first two tenets. The third tenet, heritability, he surmised that many of these traits were heritable based on the similarity between parents and their offspring. But what was missing from his theory was the mechanism, how traits are inherited. But of course, in Darwin's time, this was unknowable, right? This was 50 years before Mendel's experiments in pea plants were rediscovered, and still 50 years even before the discovery of the three-dimensional structure of DNA. So today what I wanted to do is start with a brief anecdote that links Darwin to that second great discovery of the structure of DNA. So what you're looking at here, I don't expect you to read, but maybe just to appreciate maybe a few oohs and ahs, because what you're looking at here is Darwin's last publication, published in 1882 in a little-known journal called Nature, which of course is one of our preeminent journals today. And this short article is entitled On the Dispersal of Freshwater Bivalves. And really what it is a short natural history note reporting a natural history specimen of a freshwater cockle clamped to the leg of a beetle. Why was this published in Nature even back in 1882? Well, it turns out this solved a great mystery of how these cockles could move from lake to lake among the British Midlands. But that's an aside. The relevant part to today's story is how Darwin got his hand on that particular unique specimen. Well, it was sent to him by a young shoemaker living in the British Midlands, who was a budding naturalist. He sent the specimen. They had a flurry of correspondence. And it turns out this young shoemaker's name was Walter Drawbridge Crick, which may ring a bell because nearly a century later, that young shoemaker's grandson, with his colleague Jim Watson, based on key insights made by Rosalind Franklin, made that second great discovery, that is the three-dimensional structure of DNA, and in many senses filling in that missing piece of Darwin's great theory, the mechanism by which traits could be inherited, what Jim Watson humbly referred to as the secret to life. And it's in this DNA code that we find even more evidence for Darwin's great theory, our 3 billion year existence, the shared evolutionary ancestry of all living things, as well as the subject of what I want to talk to you about today, the mechanistic details which gives rise to variation among species. So what are the precise DNA base pair changes that occur in this 3 billion base pair, at least in mammals, genome sequence? So like Darwin, the question we're interested in is how did all this variation arise? But thanks to Watson, Crick, and Franklin, we can look for at least some of those answers in the genetic code. So today I'm going to focus both on why and how traits vary in nature. So I'm going to tell you a three-part story. The first part involves, and all of which involves the linkages between gene, traits, and the environment. And as promised, the trait I'm going to focus on today is color. So first, I'm going to take you out into the field and ask the question of why color varies and how that might be determined by the environment. And then I'll take you from the field into the laboratory, where we'll ask about how color varies from a genetic perspective. And then once we've made these linkages, so here's where things get really exciting, at least from my perspective, is we can go back in the wild and ask how these traits may have evolved in other populations or in other species. So why study color? Well, color is one of the most diverse characters that we see among organisms. It can vary between species, but also within species. We know that color is involved in a number of biological processes, whether that's involved in mate choice, like the showy color of petals used to attract pollinators, or the canonical example of the male's peacock tail that he shows to attract females. It can also be used as a warning color. So this is a poison arrow frog, which is advertising its toxicity to any potential predators. And of course, no good biological story is complete without cheaters. So we have those species that mimic those bright coloration without themselves being toxic, but gain the advantage of signaling toxicity. And probably most commonly is the use of color for camouflage or crypsis. While these are all classic stories, let me just remind you that we're still discovering the uses of color in many organisms. So for example, it's been debated for decades the utility of stripes in zebras, and recent studies have suggested a relatively new hypothesis that the stripes are used to reduce parasite loads. That is, these blood-sucking flies that actually prefer to land on solid colors as opposed to striped colors. And what we used to think of the utility of changing color in chameleons, thought to be used for camouflage, is now thought to be an indicator of physiological condition. So we're still discovering new uses of color every day. Just as there are many uses of colors, there's lots of ways color is made. While we tend to think of pigments, color can also be made through structures. So this beautiful blue color in this morpho butterfly or the bright rings on this particular octopus are not made by pigments, but instead microstructures that are found, for example, in the scales of butterflies or on the feathers of birds that either reflect or scatter light at particular wavelengths that give rise to these spectacular colors. When we think about pigments, pigments can come from many places, including acquired from the environment. So plants produce carotenoids that are then eaten by organisms that give rise to, for example, this beautiful yellow color of this North American goldfinch, or the rosy color of the pink flamingo. But by and large, color is also synthesized in many organisms. So here, for example, are terran pigments that are shown in this British butterfly that are both this white color or a different flavor of terran makes this orange color. And of course, in mammals, we produce melanin that gives rise to this grizzled color that we see in the North American grizzly bear. Now, of all of these types of ways to make color, we know the most about melanin synthesis, and that's in part because geneticists have been studying color variation in laboratory mice for over a century. So many geneticists who work on mice have large colonies of mice, and every once in a while a spontaneous mutation arises, giving rise to a mouse that's a slightly different color, and using forward genetic mapping approaches, been able to identify hundreds of genes that are involved in the pigmentation process. And we don't just know the names of these genes, we have to know how they function and how they interact to produce pigmentation. But if we're interested in studying natural variation in pigmentation, laboratory mice don't make the best model, and even their wild counterparts, house mice, as many of you are familiar with or all too familiar with, they actually don't vary that much in color in their natural environments. So we've turned to a model system that takes advantage of this information, but also varies in the wild. So let me introduce you to the main actor of today's story. This is the old field mouse, known as Peromyscus polyanotus, which is often referred to as the old field mouse. And that's because these mice live in old field habitats like this throughout the range in the southeastern US. So we have Georgia, Alabama, South Carolina, and northern Florida. So throughout the range, they live exclusively in these old field habitats, similar to overgrown agricultural fields with this dark, loamy soil. And throughout the range, the mouse looks like this. To many of you, a typical mouse, a dark brown coat, a light gray belly, and a nicely striped tail. But what became interesting to us about this particular species is they also occupy these beach habitats, either the sandy dunes on the coast or these barrier islands off the Gulf Coast of Florida, as well as the Atlantic coast of Florida. And each one of these numbers represents a different subspecies of what I'll refer to as beach mice, because these mice actually live on these beautiful white sand beaches. I'm going to start by talking about one subspecies, and that one is labeled here, number 3, the Santa Rosa Island beach mouse. So these beach mice live in the most beautiful habitat. If you've been to the beaches of Florida, I don't know, it's almost like walking on hills of granulated sugar. So you can see from this habitat, which is actually one of our field sites, that we like to visit, let's say in February when it gets miserable and icy in Cambridge, Massachusetts. And here's probably one of my postdocs taking a little break from field work. But what you can see from this photo is the habitat varies tremendously, and in two important ways. First, the color of the substrate. So this is sand that, as I mentioned, is very white in color. And second, these mice experience high levels of predation because there's much less vegetative cover. So they're constantly aware of things like owls, herons, and hawks, and foxes, coyotes, and cats. And I'll tell you more about them in a minute. So it may not be surprising that when we go out in the field and trap these mice, they look different. And I should note this is not to scale, either relative or in real life. So these mice are about the size of a small ping pong ball. Okay? And what you can see from this beach mouse is that it lacks pigment on its face, on its side, and it's also missing that striped tail. Now, given that we know something about the geological history of these beaches and barrier islands, they're only about 1,000 years old. So it's very possible that this difference in color has evolved in just a few thousand years. Okay, so I know that you're all thinking, I hope you're all thinking, that this makes perfect sense, right? If you're a little mouse running around on these beautiful white sand beaches, it makes sense to have light coat color because then you're more camouflaged from those visually hunting predators that I showed you before. Well, that's a wonderful hypothesis, but of course, as a scientist, we want to be careful not to make up just those stories like those of Rudyard Kipling in 1902, how the camel got his hump, or how the leopard got his spot, or how the mouse got his fur color. And instead, what we want to do is design an experiment. So if we really want to know why color varies, we have to think about this in an experimental context. Okay, so what kind of experiment could we do? So here's my dream experiment. If we could go out and catch 100 dark mice and 100 light mice, set up enclosures on dark soil and some other enclosures on light soil, and then seed each of those enclosures with equal frequencies of dark and light mice. They're open to predators. And then we come back, let's say a month later, and see who survives. Right? On the dark soil, we'd expect dark mice to survive if color matters for camouflage. On light soil, light mice should survive. Well, it turns out, right, you can imagine that's a very complicated experiment to do, whether it's permitting or building enclosures or catching all those mice. So we initially did an experiment, which I would argue is the next best thing, and that is we made mice. So my postdoc here is making hundreds of plasticine models of mice. What's wonderful about these mice is that they look the same, they smell the same, they're the same size and shape, but she painted half of them dark and half of them light. She then released these mice with the help of some Harvard undergraduates on light soil and dark soil in random order, but in equal frequency, and then would come back the next morning and ask if there was any evidence of attack. Could we fool the predators to think these are real mice? Well, indeed we did, and here's one of our data points. So here's a dark mouse, here's its head, here's its tail, and you might notice it's missing a chunk out of its back and part of its left ear. We can even tell what type of predator this was. This was an avian attack. There's a nice bill imprint and no mammalian footprints around. And so we could count up the number of attacks in both soil types on both color variants. And here's what we found. So these charts are just showing you the relative rate of attack. So what you can notice is that if you're cryptic, that is, you match your habitat, you have a lower attack rate compared to those who are non-cryptic. And interestingly, it's equal, that is, it's equally good to be a light mouse on light soil as it is to be a dark mouse on dark soil. And this is really a nice, very simple demonstration of how color could matter to these mice. But remember that experiment I told you before about building large enclosures and seeding them with real mice? Well, we did that too. I'm just not going to go into the details of this. This is just a write-up in The Atlantic. So a couple of years ago, we made these big enclosures, released equal frequencies of mice, and it turns out it matters for plasticine mice, and it also matters for real mice color in terms of survival. Okay, so now that we have some inkling about why color matters in this particular context, the next thing I want to do, and implicated a role for natural selection, is to take you into the lab and ask how. So here's where I'm going to skip over almost a decade worth of work and get you right to the result. So here, this is our genome of our beach mice. Here are all these chromosomes with markers. But the main point I want to make is we found 3 regions in the genome that seem to be correlated with differences in color. And today I'm going to tell you about one of them, this region here on chromosome 2. Well, it turns out that region on chromosome 2 contains a pigment gene, and it's a gene called the melanocortin-1 receptor. And the melanocortin-1 receptor is a transmembrane protein, so here I'm showing you all the amino acids that are highlighted here, and this was a really enticing candidate gene when we first uncovered this region. So the first thing we did was sequence this gene. I call it the dream gene. It's about 1,000 base pairs in length. It's got no introns. It's super simple, very well conserved. And in this gene, when we sequenced it in our dark mainland mice and compared that sequence, that DNA sequence to our light beach mice, we found a mutation. Only one mutation that's highlighted here in red, and that mutation coded for a difference in amino acid. So whereas all the dark mainland mice had an arginine at this particular site, site 65, all the beach mice, Santa Rosa Island beach mice, had a cysteine. So that's a really enticing pattern, but it's just a statistical correlation. What we wanted to do was to test if this mutation actually has an effect on the receptor function and ultimately color. So to do that, I need to tell you a little bit about how pigment is made in mammals. So pigments come from these pigment cells called melanocytes. And if you don't have melanocytes, or your melanocytes don't mature, or they're slowly dying, for example, as we get older, and the stem cell population doesn't replenish them quite as quickly, that results in hair that is either white or gray, okay? Some of you may be familiar with this. I'm growing increasingly familiar with this, okay? So pigment cells are necessary for the production of pigments, but we also have discovered that spatial-specific repression of pigment cells can give rise to interesting color patterns, like these beautiful white racing stripes down the back of this African striped mouse is due to a spatial-specific delay in maturation of these pigment cells. Okay, but that's an aside. Let's say your pigment cells are working fine. You have plenty of them. They're producing pigments. In mammals, we produce two types of pigment. We produce eumelanin, or dark pigment, so that's responsible for brown to black, or pheomelanin, blonde to red. So you can look at your hair and ask whether you have eumelanin or pheomelanin, or what you used to have, right? And whether it's brown to black or blonde to red, this has to do with the amount of pigment increases the intensity of that color. And any one pigmentation cell can produce both types of pigment and switch between production of two. And so what this means is that you can get hairs like this one from a standard mouse that have this banding pattern. That is, it produces dark pigment and then switches to light pigment and then back to dark pigment again. And you can get all sorts of really cool patterns. For example, how long you produce pheomelanin as that hair is growing determines the width of that band. And there are times you can produce no pigment and get white stripes. So if you go home and you have a cat or a dog, you can start to look at their hair. I've spent a lot of time doing this. And you can see all the interesting patterns because humans, relatively speaking, are quite boring. Okay. So how does this relate then to MC1R? Well, it turns out that this one receptor is the key switch that sits on the membrane of these melanocytes and tells the cell whether to produce dark pigment or light pigment. So when MC1R is activated, it takes that extracellular signal, transfers it into the cell through the signaling molecule called cyclic AMP. When you have lots of cyclic AMP, you produce dark pigment. If it's not activated or even repressed, intracellular levels of cyclic AMP go down, and you produce light pigment. So based on this, we could make a hypothesis about what's going on in these light-colored beach mice, and that is, we thought, well, maybe this variant of the MC1R receptor has lower receptor activity. And that was something we could test in the lab, because we could express this protein in cell culture and the two variants of the protein and compare them to see if they have differences in activity. So that's what we did. So here what you're looking at is increasing amounts of activator, the thing that turns on MC1R. We measured the amount of cyclic AMP in response. You can think of this as a proxy for receptor activity. And what you get is this nice sigmoidal response curve. And this is actually data from the dark allele from the mainland mouse. We could do this same experiment. The only difference is this one amino acid change. And it turns out when you do this, no matter how much activator you give it, it has much less activity. So this is consistent with this mutation having an effect on receptor activity. And that receptor activity, then we know, has an effect on pigment and ultimately, in this case, survival of the mice. So in this sense, we were able to make this connection between gene, color, and environment and get a much more complete picture of the adaptive process. And this is really exciting because it was so complete. Sorry, I'm being slightly immodest here. But this story has made its way into evolution and biology textbooks and even the popular press. But what I'm most sort of excited about is we've teamed up with folks who are using this as a model of evolution by natural selection and making simulations that are being used in high school classrooms. So here what you can see is you can make all sorts of changes about mutations and inheritance and sex ratios. And we can simulate changes in allele frequency, whether you have a dark allele or light allele, depending on whether your predators are hunting mice on dark or light backgrounds. So for example, here are our predators. They're picking off those light-colored mice. And you can see the dark allele is going up in frequency over time. But whoop, we switched the environmental type. Now they're picking off dark mice preferentially. And you can see the light allele increasing in time. OK. So I've told you this story in this one Santa Rosa Island beach mouse about the role of this particular mutation. But if you recall, we also have beach mice on the Atlantic coast of Florida. And the question was, do these mice have similar colors? And if so, are they caused by a similar mutation or the same mutation? So just by way of reminder, here's our Gulf Coast beach mice. Here are our Atlantic coast beach mice. They're separated by about 1,000 kilometers, and they're thought to have evolved independently. So everything I've told you about is this one population, and now we're going to ask about what's going on in these populations. So we set off to the field again, probably February again, to the Atlantic coast. We caught a bunch of mice. Here we are recording their coat color pattern. And here's the best part, is that we get to release these mice. And the mice gain a little ear tag so we can follow them and look at their survival. And in return, they donate a little notch of their ear or a little tip of their tail for DNA analysis. So here's what we found in cartoon form. So there's several things I want you to note from this figure. First, all of the mice that live on beaches are lighter in color than the mainland mouse. Second, that within any one subspecies or population, there's very little variation in color. Second, by contrast, there's a lot of variation between the subspecies. But most interestingly is that when you compare the color and patterns from the Gulf Coast mice to the Atlantic Coast mice, what you see are these mice are indistinguishable, even to a trained beach mouse expert. I can't tell these apart, I can't tell these apart, and I can't tell these apart. So this suggests that very similar colors and patterns have evolved on both coasts. And now the big question is it caused by the same mutation? So we went back to our favorite gene, the melanocortin-1 receptor, and now the job was really easy. We didn't even have to sequence the whole gene. We could just genotype this one site. And it turns out that we looked at hundreds of mice from the Atlantic coast, and not one of them had the cysteine mutation. So in other words, they have very remarkably similar coat colors, but it's not caused by the same mutation. Then we went back and sequenced the whole protein, because it could be that there's another mutation in this gene that causes the same effect on receptor function, leading to light coloration, and there were no new mutations. Okay? So in this case, it looks like they're completely different mutations that give rise to differences in color. Okay? Okay, for the last thing I want to tell you about, we've identified this mutation, as I mentioned, in the Gulf Coast beach mice. It's not present in Atlantic Coast beach mice, but what about other species? Okay, so here's where I need a little audience participation. I want to ask if you can think of a mammal that is most different morphologically from a mouse. Whale's a good guess, not what I'm looking for though. Elephant, excellent. Not quite, but close enough because you guys wouldn't guess this, but it's a mammoth. Okay, so I want to tell you a story about a mammoth. So I went to a meeting, and I was sitting there. We had just found this MC1R result. I was all excited to share it, and I sat down next to a colleague who was working in Leipzig doing ancient DNA work. And he was working on woolly mammoths because, one, they were pretty prevalent. Two, their DNA had been preserved for only 14,000 years in the permafrost, like keeping DNA in a giant freezer. So this is a great species to start doing ancient DNA work. And they had started sequencing a bunch of genes. And it turns out they sequenced the melanocortin-1 receptor. So I was sitting there, and I said, we've got this great mutation in MC1R. And he said, we found a mutation in MC1R too. So we skipped a bunch of the meeting and compared DNA sequences, and it turns out that of the 3 specimens, that's 6 alleles, 1 allele had a mutation that was the arginine to cysteine mutation at position 65, which suggests that woolly mammoths, like beach mice, may have been polymorphic in color too. Now, of course, we can't do the experiments of why they might be different in color, but when these papers were published, it didn't stop the press from hypothesizing with titles like "Blonde Mammoths Have More Fun." I'll just leave it there. So what I've told you here is a story about how within a single species, you have this remarkable convergence in color caused by, at least partially, by different genes. I do want to tell you that we just published a paper looking at another gene that also contributes to color differences. It's a gene that actually physically interacts with MC1R, and there it's the same gene and same mutation that contributes to these color differences. But that's an aside, back to MC1R. Yet in very, very different mammals, we have a case where you have the same gene and the same mutation. But it turns out that MC1R is really a hotbed or a hotspot of evolutionary change because there are lots of different mutations in this one receptor that can lead to color differences. So for example, it contributes to differences in domesticated species like cattle, whether you have, I don't know what strain this is of cattle, but a red or a black form. If you own a Labrador retriever, whether you have a blonde lab or a black lab, this is caused by a single mutation in MC1R. Even in lizards, lizards that live on the white sand dunes in New Mexico, that's this one up here, relative to its wild type, it's caused by a different mutation in MC1R. And even we have mutations in MC1R, mutations that cause similar changes to receptor function, and in this case, particularly high frequency in some populations, like Irish populations. And it turned out my colleague who was working on ancient DNA in mammoths also has worked on Neanderthals. And it turns out there's a mutation in MC1R, different from what we find in humans, so it's not contamination, that has a similar effect on receptor function, and we now know about 6 to 10% of Neanderthals likely had red hair as well. So with that, I hope to have convinced you that we can start to disentangle both how and why organisms can adapt to novel environments, becoming increasingly important as our environment is constantly and more rapidly changing. So with that, let me say that I hope by telling you about experiments that were right up Darwin's alley, that is simple experiments done in the field combined with genetic analysis, we're starting to untangle what Darwin referred to as that perfection of structure and co-adaptation, which most justly excites our admiration. So with that, I thank you for your attention, and let me end in case you were intrigued by the first image and wanted to know what they're from. Here are the answers to that quiz. Thank you.
Well, my name's Richard Lounsbery, and I'm the Dean of the Faculty of Natural Sciences, and I'd like to thank Hopi very much for that. But I'd also like to ask you if you've got any questions. We've got 10 minutes. Questions? Go ahead.
I was just wondering, have you looked at epigenetics in colour change?
That's a great question. The gene that I just briefly referred to at the end that has the same mutation in both Atlantic and Gulf Coast beach mice is a gene called agouti, and I sort of think of agouti as the poster child for some of the first evidence of epigenetic change. So what I'll say is that in doing, for example, our genetic crosses, we haven't found anything that's inconsistent with simple Mendelian inheritance, which doesn't mean that epigenetics isn't involved. And in fact, epigenetics, the thinking of epigenetics played an important role in the establishment of these mice, because initially it was thought these beach mice were on the beaches where there was increased humidity, which in turn led to color changes. That was the initial hypothesis. And it wasn't until they brought them in the lab, raised them in a common environment, and the colors stayed the same that they realized there wasn't any of environmentally induced change. Again, it doesn't mean it doesn't play a role, but we haven't found any evidence for it or a need to invoke it.
Okay. Dan?
You mentioned briefly, we know a bit about the how people have different hair color. Do we know is there anything about why people have different hair color?
There's a lot of wonderful speculation about this, which I am loath to step into, but sexual selection is one that's been invoked many times, that red hair, light skin was thought to be beautiful at one point. It's a tricky literature, but I'll point you to that and stay clear of it myself.
Okay, another question? Oh, down here.
Well, I've got a question as a non-scientific person. I'm an engineer, not a bio person. You talked about animals.
Yep.
What about plants?
Yes.
Is that applied to plants? I mean, the leaf is so variable.
Yes.
What's the magic ingredient?
Yeah, so these questions about, why things are the way they are, why do leaves have so many different shapes, or why do mice have many colors and so forth, or why do humans have so many colors, I think are in my opinion, much harder questions to answer than the how questions. So let me just say that. There have been similar lines of research done in plants, often focused on the pigmentation that occurs in the petals, in part for similar reasons here. Those pathways have been really well worked out, and so you sort of have a starting foundational understanding of how petal color may be made, and then you have a nice set of candidate genes, and you can start to, in terms of the, why there's so many different colors, people have done beautiful, elegant experiments, and I have to say in plants, I get very jealous because you can do one of these enclosure experiments in plants, and the plants stay still, whereas the mice, right, will do anything and everything to try to get out of these enclosures, and have shown they have effects on attracting pollinators, and there's differences when you have two plants that are in sympatry versus allopatry and so forth. So there is really beautiful work on plants. It just isn't, I don't know as much about it.
Sure, thank you for the interesting talk first. Thank you very much.
Okay, any other questions? One final question? Okay. Crystal.
Thanks, Hopi. I thought that the reproducibility that you got in the variation in the Gulf Coast and the Atlantic Coast was really interesting. Do you know if that is related to a restriction as to kind of the variability that you can actually get in color, or is it maybe predator discrimination and their ability to actually determine?
That's a great question. And again, it's kind of one of these why questions. What I can tell you is that what we've looked at is in the Gulf Coast, the populations furthest to the west are near where the Mississippi comes out, and there's a lot of drainage, and so the soil is actually a little bit darker because it's got sort of actual soil from the Mississippi that comes there, and then some of the beaches are much more pristine, white, and there's variation. It turns out the mouse coat color, just the reflectance of their coats, not even the pattern, matches that trend. Similarly, the mice that are on the beaches on the Atlantic coast that are most north, there's a lot of coral there, so the soil is a little bit more pinkish, and the reflectance is a little bit different in those mice. So that's the extent to which we've looked at correlations with environment, but I think the question of genetic constraint is a really interesting one. So one of the reasons I think MC1R is sort of this hotspot of evolutionary change is that it's at the very end of the pathway. So if you mess with MC1R, you're not messing with a lot of other things. There's a little caveat detail to that, but I'll leave that out for now. Whereas other genes that are important for pigmentation that are more higher in the pathway also can affect, have these deleterious effects on other aspects. And it's a short protein, very simple regulation, and there's apparently lots of ways you can affect that receptor function and tweak it so that you don't wipe out color completely, but that you can just modify color slightly. We've looked at that gene the most. Other genes, like this agouti gene that I referred to, much more complicated genes, does have pleiotropic, can have pleiotropic effects, and so maybe that's a little bit more constrained. But my guess is that genetic constraint, as well as these ecological factors together, sort of limit what we see. But wouldn't it be great if we could, speed up evolution and watch things over time to see what's possible and what that adaptive landscape looks like? That's why I'm always a little jealous of experimental evolution in microbes, because you can do those types of experiments within your lifetime.
And there's one final question, young lady there.
You did mention that there was like 2 different beach mice, and they had a similar color change, but different like genes that caused that. What is there a particular difference between the two different genes that cause the color change?
Yeah. So what I would say is that I think of these, the Atlantic coast and the Gulf Coast, as two independent evolutionary experiments that have played out over, we think, about 8,000 years. 3 to 8,000 years. And so what we think has gone on is just by chance, the populations on the Gulf Coast, that mutation appeared. And that then made its way to the beaches where it was beneficial and spread on the beaches. And that mutation just never occurred on the Atlantic coast. The second gene that I didn't tell you about, this agouti gene, looks like it's a mutation that already was segregating at very low frequency in the mainland population, where it would be selected against, so that's why it's at low frequency, because you don't want to be light color on dark soil either, but then made its way both to the Atlantic coast and to the Gulf Coast and selected up. And that mutation sort of is very old relative to MC1R mutation, which looks to be very young. So I think the difference is just because evolution works with the mutations that it has, and in one case it had that MC1R mutation, and the other it didn't.
Okay, I'm now going to hand over to Vincent, my colleague, who's going to give a vote of thanks. Oops.
Yes, so we have a tradition in the college somebody would give the vote of thanks. So it's my great pleasure and honor to be able to give the vote of thanks to Hopi. I've known Hopi for some years now, and I have followed her work for many years, of course, and I think she had an incredible career. But what really is impressive is she was always ahead of us on so many topics, and she turned on its head so many widely held beliefs. So, for example, 20 years ago, she wrote a seminal paper on chromosome evolution. And so mammals, we normally think male would be XY and female will be XX. And Hopi challenged his view and published a paper where she showed that XY female evolved multiple times in South America in some group of mice. One of her most highly cited papers, she actually wrote an account how mutation in cis regulatory elements and protein coding genes would be underlying the evolution of morphology, physiology, and also complex traits like behavior. And that was a vision that she put in this paper that we've all followed ever since. And in fact, to help us following this vision, she even invented a new technique called RAD, where we could sequence a small portion of the genome at a very low cost. And the paper was published, and it was just mad inside the community. Everybody started to do this RAD sequencing. We're still doing it, and this was a revolution in ecology and evolution thanks to Hopi. She was also been very active in big debates. So, for example, in 2014, there was a movement of some scientists who wanted to create or were calling for a new theory of evolution, something called a new extended evolution synthesis. And Hopi was one of the very few people who said, no, we don't need any of these. And again, a few years after, we realized that she was right. And although it's something still debated, there was a paper in The Guardian not long ago, but Hopi was already there telling the truth many years before everybody. I think her big thing now, she hasn't discussed, is to show how small genetic change can affect the whole of the behavior, so something a lot more complicated than color, and there's amazing papers that have come out recently. Just to finish, I know some of the postdocs that are working with Hopi, and they all, I contacted them, and they all praised how wonderful mentor she was. And indeed, many of these postdocs went on brilliant careers like Hopi. Some have been editors at Nature, Science, or got professorship at Columbia or Princeton. So I think you will all agree she had an amazing career. The talk was absolutely fantastic and an inspiration. So thank you again very much for coming to visit us in London.